Hey folks, what we're going to talk about in the next 3 or 4 videos is this complex process called DNA replication, right? If you think about it, before any cell divides, it has to completely and faithfully replicate its entire genome so that both daughter cells have the complete full set. This is really fundamental to all of life. It's part of what we think of as the central dogma of biology, DNA replication and gene expression through transcription and translation. We'll talk about those processes a little bit later, but right now we want to talk about how the DNA gets replicated. So in this first video clip, what we'll do is we'll take some overview perspectives and try to understand what the genome looks like a little bit, remind ourselves of some of those details of how DNA is structured and so on. We're going to talk then about how DNA gets packaged, because we've got these very long molecules that are packed very tightly into every single nucleus of every single cell with only a few exceptions. And then we're going to look at a model for replication called the semi-conservative model that will give us, again, some perspective on how this process is happening. Then we'll stop this video, and in a new video we'll look at the actual molecular mechanism of DNA replication. What are all the details, all the little proteins, all the steps involved? And that's a video you're going to want to watch multiple times until it really makes sense to you and you can track with all of those steps, even to the point of where you could, without any aids, sit down and talk someone else through that entire mechanism step by step and all the key proteins and molecules involved in that. So let's start with an overview at this point. Let's remind ourselves of how the genome is organized. The term genome refers to the collection of all the genes in any living cell, right? And in a multicellular organism like you and me, every single one of your cells has the exact same genome. The genome doesn't vary between a skin cell and a liver cell. The collection of genes is exactly the same. What varies is which genes are being expressed in a skin cell versus what genes are being expressed in a liver cell. So the genome refers to all of the genes. When we use the term chromosomes, we're talking about an individual molecule of DNA that is discrete and distinct from other potential molecules of DNA that has multiple genes coded on it. So what do we mean by genes then? A gene refers to a single unit of DNA from an informational standpoint. A chromosome refers to a molecule, right? A gene refers to a unit of information, and we're going to see when we get into gene expression that there are sequences of A's and T's and G's and C's that say start here and stop here. There's no physical break between those genes. There's simply an informational break and a start and a stop. And so one gene is going to code for a protein. Remember, a gene is a recipe. And the gene is the recipe for exactly how to put together a protein, which will then be polymerized, folded, and sent off into the world to do whatever it does and give the characteristics intended to that particular cell type. So any given chromosome is going to have multiple genes. For example, in a human chromosome, you're going to have thousands of genes per chromosome. And then the nucleotides are the actual chemical subunits that we know get polymerized through covalent bonding along this sugar phosphate backbone that actually create the genes, the informational units. So nucleotides are sort of the raw materials for building genes. Multiple genes are found on any given chromosome, and the collection of all the genes, whether that's a single chromosome or multiple chromosomes, is what we call the genome of an organism. Now, I said that informationally a gene starts and stops based on sequence. Here's an example, right? I just pulled up a little bit of sequence from a plasmid called PAKD4. A plasmid is like a mini chromosome that bacteria can carry. In fact, in nature, more bacteria carry plasmids than not. Not essential for life functions under most conditions. You could remove the plasmid and the bacterium could survive as long as conditions were relatively favorable. But most bacteria carry these little tiny miniature circular chromosomes. So here's a portion of the sequence where each of the letters represents one of those chemicals that we call a nucleotide. Right, so these chemicals, these nucleotides, are polymerized together and held together through covalent bonds. And then I highlighted in red all the nucleotides of one particular gene called a transposase gene that's important for transposons, these horizontal gene transfer mechanisms of jumping genes. And what you can see is that there's no break, there's no physical break. It's simply informational that says start here and stop here. So keep that in mind as we're talking about what genes are and what chromosomes are and what genomes are. All this information needs to be duplicated in an exact replicate molecule prior to cell division. Here's another example, just to help your scientific imagination wrap your brain around what a chromosome looks like. In this case, this is a plasmid, one of these little miniature chromosomes, PAKD4, the one that I pulled that transposase gene off of. 56,803 base pairs, right? Double-stranded DNA twisted into a double helix. In the case of a plasmid, it's covalently closed circles, so there's not a 5' end and a 3' end, though there's still directionality to both strands, and the strands are in reverse complementary orientation, like we've talked about. You can see that each of those colored blocks, they're sort of like chubby arrows, represents a different and unique gene. Different sizes to the genes, and you notice there are arrows pointing in different directions because the genes can be coded on one strand or the other and therefore pointing in one direction or another. So on a plasmid, for example, you might find 30, 40, 50 genes. A chromosome, a bacterial chromosome, you're going to find 4, 5, 6,000 genes because bacteria typically only have a single chromosome, so their entire set of essential required genes are going to be found on that chromosome. Let's not forget how DNA is structured. We talked about this earlier in the semester. Nucleotides make up the basic structure of DNA. They're polymerized via sugar phosphate linkages, right? So you've got this backbone that goes from phosphate to sugar to phosphate to sugar to phosphate to sugar and on and on and on. But there's no information in that because it's just alternating, right? Sugar, phosphate, sugar, phosphate. Where the uniqueness, where the information comes from, is each of those nitrogenous bases that's kind of hanging out there in the breeze, the A's and C's and T's and G's. And remember that each of the two strands is in reverse orientation, so you can see in this image here they're flip-flop directionally relative to one another. Keep track of 5' and 3' terminology. That becomes really important when we look at the mechanism of DNA replication. And then we get hydrogen bonding following the base pairing rule. So A's and T's like to hydrogen bond with one another. They form 2 hydrogen bonds. G's and C's comfortably form 3 hydrogen bonds with one another. Individually, those hydrogen bonds are relatively weak, but when you've got thousands or millions of these, they can hold the DNA together relatively well. And then I want to talk about DNA packing, right? It's each of these chromosomes, let's say in a human, for example, a chromosome averages 6 to 8 centimeters long if it was just stretched out. How the heck do you fit 46 of those in every single nucleus of all of your cells, with a couple exceptions of blood cells that don't have them? It's through packing, right? They're not just linearly stretched out. They're packed very intentionally and very tightly so that they can fit into the nuclei. So let's start with these DNA histone complexes called nucleosomes. Histones are proteins that wrap around DNA or allow DNA to wrap around them about every 20 to 100 base pairs. So there's a stretch of DNA that has 20 to 100 base pairs that's not contacting a histone protein. And then there's about 146 base pairs actually wrapped around the histone protein itself, forming what's called a nucleosome. Even though that DNA is physically contacting these proteins, it's still accessible for replication and for transcription and ultimately translation. And so these nucleosomes aren't somehow packing the DNA away in a manner that takes them out of the picture. That DNA is still very much accessible by the machinery of replication and transcription. So what you end up with from these nucleosomes is what's called sometimes beads on a string. right? This 30 nanometer fiber that it's called 30 nanometers because it takes on a width of about 30 nanometers. So it starts getting wider, right? The original double helix is about 2 nanometers. It starts getting wider now, 15 times wider, 30 nanometers, but now it's getting shorter, and that's the price that we pay for compacting it. Now, that's not all we do. We don't just form these nucleosomes into this 30 nanometer fiber. We also loop them onto fibers of protein in the nuclear matrix. Nuclear matrix proteins are kind of like a cytoskeleton inside the nucleus, and it forms a scaffolding to loop this 30 nanometer fiber and attach it. And again, we get wider. Now we're around 300 nanometers, but we also get a whole lot shorter. Each of these loops is somewhere between 25,000 and 200,000 base pairs. So in a eukaryote, we don't get all this extensive packing in prokaryotes. But in a eukaryote, we've all of a sudden taken this really narrow molecule of 2 nanometers. We've made it a lot shorter and a lot chubbier in the process. This stage of radial loop domains, we call this euchromatin. Eu meaning true or real. Chromatin. Chromatin is the term for the DNA when it's packaged inside the nucleus. And euchromatin is that DNA that is still fully accessible for gene expression. Each of the chromosomes finds itself in what's called a chromosome territory, a general region that you would find, for example, chromosome 1 or chromosome 2 in a human nucleus. And that's determined by where in the nuclear matrix it's bound. We can then wrap that and loop that even tighter into what's called heterochromatin. The heterochromatin is typically not accessible. Okay, euchromatin is accessible for gene expression. Heterochromatin is essentially packaged too tightly for enzymes to get in and read anything. So heterochromatin is a way for a cell to shut down certain regions of its chromosome, but also to package it even tighter and even wider, ultimately for the process of cell division. So ultimately what you see here is the metaphase chromosome that's 1.5 micrometers. That's the size of an E. coli. All the way across, this is the typical chromosome we think of that's already been duplicated, so it has that X-looking pattern because there's 2 of them linked together at a centromere. And they're so short and so dense that with the proper staining techniques you can actually see them in a light microscope. And that's what the term chromosome means, visible body. Chromo for visible and zome meaning just a structure or a body. So the DNA gets packed really densely through each of these kind of levels of packing. If it's in the radial loop domains, we call it euchromatin, and it's accessible for gene expression. If it's packaged even further, if it's a region of a chromosome that's packaged further, or the entire chromosome packaged further, we call that heterochromatin. And at least temporarily, if not permanently, those genes are off limits for gene expression. Now I said DNA replication has to precede division. Hopefully that's obvious why. In eukaryotes, that division is going to come primarily in the form of mitosis, but also potentially meiosis if we're talking about the formation of sperm and eggs. In prokaryotes, that cell division happens by binary fission. In either case, you have to start the process with faithful DNA replication. Otherwise, you're going to give one daughter cell mutated DNA or no DNA at all. And that's bad news for the survival of the organism. Now, I mentioned this semi-conservative model of DNA replication. It's this idea, semi-conservative, has nothing to do with politics, so get that out of your head, right? It doesn't mean Hillary Clinton and Donald Trump. No, semi-conservative model of replication means your parent molecule, which consists of 2 reverse complementary strands of DNA, in the 2 new daughter molecules, half will be the original parent molecule and half will be a brand new molecule. So the first step is to separate these 2 molecules from each other so that they can become the template. They can provide the instructions for what new nucleotides go where, just based on the base pairing rules. So if you separate these 2, then what you see in blue are the original parent strands. Fill in the new daughter strands with new nucleotides, just following the base pairing rules, and in the end you end up with 2 new molecules that are half old, half new, in other words, semi-conservative. Conservative means something was saved. In this case, semi-half. Half the original molecule was saved in the new daughter molecules. So you have half dark blue, half light blue in the diagram here. This is what's called the semi-conservative model or idea of DNA replication. And again, the important thing is that we have to separate those strands so that they can each act as a template, providing the instructions for all the enzymatic machinery for what nucleotide to put where, simply based on the base pairing rules and that hydrogen bonding. You're going to want to take a look at the Meselson and Stahl experiments in your textbook. If you're using the Brooker textbook, it was originally on page 183, 184. I think they've moved it in the newest edition. If you're using a different textbook, just go to your index. For sure it's going to be in there. And you want to take a look at how they figured this out. Some really interesting experiments that they did to figure out exactly how this worked. So that's it for the introduction. In the next video, we're going to get started looking at the molecular mechanism itself to see exactly how this happens. Hope that was helpful for you.